Hello NEET aspirants, you are just one day close to the exam. Let us discuss all the diagrams in a very quick mode. As previously we have discussed the diagrams of biological classification, plant kingdom, morphology and anatomy. Now we will revise quickly all the diagrams of cell the unit of life, cell cycle, and then we will look into photosynthesis in higher plants, and then respiration, and then we will look into plant growth and development. Let us revise quickly all the diagrams of these concepts just within half an hour and just within 30 minutes. Let us complete all this. Okay. So, yes, if we look into the shapes of the cells, okay, this shapes of the cells diagram is important, Nana, where we may get, like in the match the following questions also we may get, like red blood cells are usually round and biconcave. Red blood cells are usually round and biconcave. White blood cells are amoeboid. Columnar epithelial cells are long and narrow, and the largest cell is nerve cell. Nerve cell is the largest cell, wherein it is branched and long. Okay, tracheid. Tracheid is enucleated. Tracheid is enucleated, and these are dead cells. Okay, they have long tube like cells with tapering ends, and at the center you can see pits or pit pairs. Okay, so tracheids are elongated cells. When it comes to mesophyll cells, okay, mesophyll cells, these are round and oval. Next, yeah, here comes the size of the cell. A eukaryotic cell is of 10 to 20 micrometers in diameter, and bacterial cell is 1 to 2 micrometers in diameter. Pleuropneumonia like organisms, mycoplasma, like these are 0.1 micrometers in diameter, and viruses are from 0.02 to 0.2 micrometers in diameter. You have to remember each and every number of this, very much important beta. Okay, and then if you look into, yeah, a eukaryotic cell, here he can give a eukaryotic cell and he can ask any of the labeling for you. This is a plant cell beta. How you can identify? By the cell wall, you can see it over here. Okay, by the cell wall you can clearly identify that this is a plant cell, and they have plasmodesmatal connections. They have plasmodesmatal connections, and you can see this as a plant cell. Even though this is given as black and white diagram, you can clearly observe, because vacuole is occupying 90% of the cell volume. Vacuole is occupying 90% of the cell volume. When it comes to the nucleus, nuclear envelope, this membrane is contiguous with endoplasmic reticulum. Most important question. Okay, nuclear membrane is contiguous with endoplasmic reticulum. Nuclear membrane contiguous with endoplasmic reticulum. If this endoplasmic reticulum has small ribosomes over it, you will call it as rough endoplasmic reticulum. This is called as rough endoplasmic reticulum. They are helpful in the synthesis of proteins. They are helpful in the synthesis of proteins, whereas smooth endoplasmic reticulum, which does not have any ribosomes over it, they will synthesize lipids. They will synthesize lipids in animal cell, though smooth endoplasmic reticulum will also synthesize steroids. Okay, and then after endoplasmic reticulum it is Golgi bodies. Okay, after endoplasmic reticulum it is Golgi bodies. Then in connection to the Golgi bodies, you will be having lysosomes and vacuole lastly, wherein all these single membrane bound organelles, endoplasmic reticulum, Golgi body, lysosomes and vacuoles, many at times asked a question about endomembrane system, that is all these 4 organelles comes under endomembrane system, whose functions are coordinated, wherein the naive proteins and lipids that are synthesized from rough endoplasmic reticulum and smooth endoplasmic reticulum, okay, and smooth endoplasmic reticulum are given to the cis phase or convex phase of the Golgi. They are given to cis phase or convex phase of Golgi, and they are released in the form of vesicles from trans phase of the Golgi bodies. Okay, these are released in the form of vesicles which are rich in the hydrolytic enzymes called as lysosomes. Lysosomes attaching to a digestive molecule or to any dead organelle or to any dead constituent of the cell, giving out so much unwanted material which is finally deposited in the vacuole. This deposition takes place in active transport. Okay. What is the membrane of the vacuole, Nana? The membrane of the vacuole is tonoplast. Tonoplast is the membrane of the vacuole. And how the materials will enter into the vacuole? How the materials will enter, beta? The materials will enter into the vacuole in an active transport method. Okay. And what are other small cells which are present? Membrane bound cells which are present in a plant cell? Those are like microbodies, peroxisomes, glyoxysomes. These are present like small membrane bound structures. And what are the non-membrane bound organelles that are present in a plant cell? Those are ribosomes. Ribosomes are non-membrane bound organelles present in the plant cell. What are double membrane bound organelles present in a plant cell? Chloroplast, mitochondria and nucleus. Fine. And what are the types of ribosomes that you can see in a plant cell? You can see 70S ribosomes in chloroplast, and mitochondria, in chloroplast and mitochondria. Okay. And you can see 80S ribosomes over rough endoplasmic reticulum and inside the cytoplasm. Okay. Keep that in your mind. Fine. Next, when it comes to animal cell, this is a villi projections. These are small microvilli projections which are microvilli, and their limiting boundary is a plasma membrane, and they have membrane bound organelles include centrioles and ribosomes. Centrioles are absent in a plant cell. Many times it is asked, beta. Centrioles are absent in a plant cell. Centrioles help in the spindle apparatus formation. These are helpful in spindle apparatus formation. Okay. So, yes, how spindle apparatus are formed in a plant cell then? Spindle apparatus are formed by a microtubular organizing complex. Here they are formed by centriole in an animal cell. Then what are the single membrane bound organelles? Of course, here also you can find the endomembrane system that includes endoplasmic reticulum, Golgi bodies, lysosomes and vacuole. Vacuole size is very limited in an animal cell. It is not like in plant cell where 90% of the cell volume is occupied with vacuole. Here in animal cell vacuole size is very limited. When it comes to double membrane bound organelles, what are the double membrane bound organelles you see here, beta? Nucleus and mitochondria, that's it. And what type of ribosomes do you find here? 70S ribosomes you can observe in mitochondria. 70S ribosomes you can observe in mitochondria, and 80S ribosomes you can see it in 80S ribosomes you can see it in the cytoplasm of an animal cell. Okay, 80S ribosome you can see it in the cytoplasm of an animal cell. Next, when it comes to the plasma membrane model, the most widely accepted plasma membrane model is what? Fluid mosaic model. The most widely accepted model is fluid mosaic model. Fluid mosaic model is given by Singer and Nicholson in the year 1972. Okay, fluid mosaic model is given by Singer and Nicholson in the year 1972. According to them, okay, how proteins, lipids and sugar molecules are present here. Okay, according to them, plasma membrane is composed of proteins, lipids and carbohydrates. The carbohydrates are just of approximately 5%. They are in the form of glycoproteins and glycolipids. Okay. If you consider a RBC cell, if you consider the plasma membrane of red blood cell, you can clearly see the lipid content of the plasma membrane in red blood cell is 52% and protein is 42%, is 40% and protein content is 52%. And here if you see lipids, most of the lipid composition in the plasma membrane is phospholipids. It is phospholipids, and you can clearly see the quasi-fluid nature of lipids. Lipids will have a hydrophilic head and a hydrophobic tail. Okay, it has a hydrophilic head and hydrophobic tail. Okay, it has a hydrophilic head and hydrophobic tail. Fine. Hydrophilic head is towards the outer side and hydrophobic tail is towards the inner side, and this lipid composition is in quasi-fluid nature. So, quasi-fluid nature of lipids, in this quasi-fluid nature of lipids, proteins will appear as icebergs in the sea. Okay, proteins are as icebergs in the sea of lipids here. So, proteins are also of three types in this plasma membrane. They are peripheral proteins, transmembrane proteins and integral proteins. Peripheral proteins are on the any of the peripheral side, that is outer surface of the lipid membrane, and integral membrane is towards the inner side of the membrane. Okay, either it can transpan two sides of the plasma membrane or only one side of the plasma membrane. Transmembrane protein is also transpanning two sides of the plasma membrane. Okay, transmembrane protein will transpan two sides of the plasma membrane. So, here what you have to remember, Nana, quasi-fluid nature of the lipids will fill these proteins, like proteins how they are present inside the plasma membrane, they are as icebergs in the sea of lipids. And how does carbohydrates present? As glycoproteins and glycolipids. These glycoproteins and glycolipids are synthesized from what organelle? Golgi bodies. All these are gunshot points, beta, just take it out. Next, here if you see the nucleus structure, labelings are very important. Okay. So, this nucleus has nuclear pores over its surface, and the extensions, the reticular extensions of it are called as rough endoplasmic reticulum, and over this rough endoplasmic reticulum you can find ribosomes present. Over this rough endoplasmic reticulum you can find ribosomes. Okay. And the smooth endoplasmic reticulum, which is at the side of rough endoplasmic reticulum, they will synthesize lipids and steroids. Okay. Next to this you can easily see the Golgi bodies. The cis phase of the Golgi is towards the endoplasmic reticulum. This cis phase is called as cis or convex phase, convex or forming phase. Okay. Cis phase, convex phase or forming phase towards the endoplasmic reticulum. Next, this concave phase, okay, this concave phase is also called as transphase, transphase or concave phase, transphase, concave phase or mature phase, okay, or mature phase. Transphase, concave phase or mature phase is towards the cytoplasm. Cis forming phase is towards the endoplasmic reticulum. Here the packing of proteins and lipids takes place, ultimately that leads to the formation of glycolipids and glycoproteins, glycolipids and glycoproteins. Okay. These two diagrams, fix it in your mind. Most importantly, look into the labelings of it, beta. And next, if you see the double membrane bound organelle, that is mitochondria. The outer membrane of mitochondria is easily permeable, where permeability is more for the outer mitochondrial membrane, whereas the inner mitochondrial membrane is selectively permeable. Inner mitochondrial membrane is selectively permeable. Okay, inner mitochondrial membrane is selectively permeable, where in inner mitochondrial membrane you can clearly see the electron carriers, okay, protein complexes are all present in the inner mitochondrial membrane only. Electron carriers and protein complexes, whichever are required for the ETS process, they are present in the inner mitochondrial membrane itself. And the inner mitochondrial membrane is into foldings, infoldings called as cristae. Okay, inner mitochondrial membrane infoldings are called as cristae. Then inside the mitochondrial matrix you can find 70S ribosomes and double stranded circular DNA. Okay. Most importantly, you have to know the locations of aerobic respiration as well beta here. Okay, locations of aerobic respiration. Consider this as outer mitochondrial membrane where the membrane is easily permeable, and this as inner mitochondrial membrane which is selectively permeable with electron carriers and protein complexes. And here this infoldings of inner mitochondrial membrane is called as cristae. Okay, and the space between outer mitochondrial membrane and inner mitochondrial membrane is called as intermembrane space, and inside is mitochondrial matrix which has 70S ribosomes and double stranded circular DNA, Clear? Fine. Now here we have to know the locations of aerobic respiration. Glycolysis which happens in the cytoplasm, where glucose is incompletely or partially oxidized to 2 molecules of pyruvic acid. They are incompletely or partially oxidized to 2 molecules of pyruvic acid. These 2 molecules of pyruvic acid will directly enter into the mitochondrial matrix. Inside mitochondrial matrix, this pyruvic acid first undergoes oxidative decarboxylation, and then after oxidative decarboxylation TCA cycle will occur, tricarboxylic acid cycle or Krebs cycle. So, what is the site of Krebs cycle? Mitochondrial matrix. What is the site of oxidative decarboxylation? Mitochondrial matrix itself. Okay. Now, the electron transport system is activated, pumping the protons from here matrix to the intermembrane space. What is the site of ETS, electron transport system? That is mitochondrial matrix, inner mitochondrial membrane. Okay. The site of electron transport system is inner mitochondrial membrane. Keep that in your mind beta. Okay. This is all about mitochondria. These are the most expected questions. Okay. And then if we look into chloroplast structure. Chloroplast has a double membrane bound organelle, outer membrane and inner membrane. Okay. Here also the outer membrane and inner membrane permeability is different, but here in chloroplast the outer membrane is not that permeable as mitochondria. Keep it in your mind. Next, the membranous sac-like structures. Inside the chloroplast is called as thylakoid. Okay, membranous sac-like structure called as thylakoid, and the stalk of the sac-like structure is called as grana. And the fluid filled space is stroma. Where inside the stroma you can find a double stranded circular DNA. Clear? Understood? And one grana is connected to other grana by stroma lamellae. This is about chloroplast. Clear? And here in the chloroplast also you should clearly know what are the sites of light reaction and dark reaction beta. So, photosynthesis, if you take photosynthesis, in photosynthesis light reactions and dark reactions will occur. Light reactions, what are the 3 consecutive steps? First is absorption of light in the form of photons. This absorption of light in the photons is taken by thylakoid membrane, that is called as thylakoid lamellae, okay, thylakoid membrane or thylakoid lamellae. Second is photolysis of water, wherein one water molecule will give rise to 2 protons plus 2 electrons plus 1 oxygen atom, okay. So, this photolysis of water will take place in thylakoid lumen, okay, this takes place in thylakoid lumen. Next, third is formation of assimilatory power, that is ATP and NADPH. This takes place in the stroma, okay. Assimilatory power is released in the stroma. Next, when it comes to light independent reactions where carbon dioxide is fixed to glucose utilizing the assimilatory power of the light reaction, this takes place in the stroma of the chloroplast. All these locations are 100% important beta, okay. And next, if you see this ribosomes, this is the larger subunit of ribosome, smaller subunit of ribosome. Here what you have to remember, the prokaryotic cells and mitochondria, inside the mitochondria and inside the chloroplast, you can visualize or you can observe 70S ribosomes. You can see 70S ribosomes, whereas in the cytoplasm of eukaryotic cells, you can observe 80S ribosomes, okay. Cytoplasm of eukaryotic cells, you can observe 80S ribosomes. And then if you see centriole structure and cilia flagella structure, that is cartwheel arrangement, okay, of cilia and flagella. This is of cilia and flagella, okay. Cilia and flagella, it is what arrangement beta? 9+2 arrangement. Cilia and flagella, it is of 9+2 arrangement. Listen keenly, where at the center you have 2 mitochondrials, that are central microtubules, okay. At the center you have 2 microtubules, you call these as 2 central microtubules, which are covered or coated by a sheath, okay. The central 2 microtubules are covered by a central sheath, and this central sheath is attached to 9 radial spokes, okay. They are attached to 9 radial spoke, where each radial spoke has 2 peripheral microtubules. So, here you have 9 radial spokes, okay, plus central 2 microtubules. So, this is what 9+2 arrangement. 9+2 arrangement you will see in cilia and flagella of eukaryotic cell, okay. Eukaryotic cilia and flagella, you can observe 9+2 arrangement. When you look into the centriole, in centriole at the center you can find the proteinaceous hub, To this proteinaceous hub, 9 radial spokes are arranged like this. To this 9 radial spokes, 3 peripheral microtubules are arranged in this way, okay. 3 peripheral microtubules are arranged in this way. So, this is 9+0 arrangement. For example, if the question is to calculate how many total microtubules are present in the eukaryotic cilia and flagella, that is having 9+2 arrangement. If that is the question, at the center you have 2 microtubules plus 9 radial spokes, each radial spoke has 2 microtubules, that is 9 into 2. Then what do you get beta? 2 plus 18, that is 20 microtubules, okay, in the eukaryotic cilia and flagella. When it comes to the total microtubules in the centriole, in the center you do not find any microtubule, in the center you can just find the proteinaceous hub. To this proteinaceous hub, 9 radial spokes are present. Each radial spoke is having 3 peripheral microtubules. In that case, 9 into 3, on total you will have 27 microtubules. Okay, on this total you have 27 microtubules, that you have to keep it in your mind. Okay, and when it is of this chromosome structure, the two disc shaped structures attaching the primary constriction. The primary constriction is centromere. Primary constriction is centromere. Attaching to this centromere, two disc shaped structures called as kinetochores. Two disc shaped structures adjoining the primary constriction, that is centromere, called as kinetochores. And there is secondary constriction above this. This secondary constriction is called as satellite DNA. This secondary constriction is what, Nana? Satellite DNA. Okay. So, when DNA is condensed to chromosome, this is a diploid chromosome where complete set of chromosome. At the center, primary constriction is centromere. Adjoining to centromere, you have disc shaped structures, that is kinetochores. Secondary constriction is called as satellite. Here this is the secondary constriction where it has repetitive DNA. It has repetitive DNA. Okay, clear? And then here if you see shapes of the chromosomes, so many times asked question. It can be diagram based, it can be in the match the following, or it can be a direct statement also. So, how you can look into the shapes of the chromosomes? First is, if the primary constriction, that is the centromere is at the middle, then you have two arms of the chromatids. Okay, two arms of the chromatids. Fine. The two arms of the chromatids are equal. Two arms of the chromatids are equal. Then during anaphase of the cell division, this forms V-shaped chromosomes. Okay. In the anaphase, centromere is close to one end where you can find one short arm and one long arm. Short arm is called as Q arm, long arm is called as P arm. Okay. This is short arm and this is long arm, Q and P. So, here this type of chromosome is called as submetacentric chromosome, and V-shaped where two equal arms of the chromatids, this is metacentric chromosome. Metacentric chromosome. Metacentric. This is submetacentric. Here you can find extremely short arm. This is extremely long arm. Okay. This is acrocentric. Okay. Acrocentric. And the last, very invisible short arm and a long arm. This is telocentric. Telocentric. Okay. Telocentric chromosome will form I shape during the anaphase, and this acrocentric will form J shape. Submetacentric will form L shape. Submetacentric will form L shape. So, these things you have to keep it in your mind with respect to shapes of the chromosomes. And now, when it comes to cell cycle has two phases. Okay, cell cycle has two phases. From this diagram you can see, one is large interphase. Interphase you will also call it as inactive phase. Interphase you will also call it as what, Nana? Inactive phase. And one more phase, that is M phase. You have two phases, interphase and M phase. Okay. Interphase has three substages. Those are G1, S and G2. Okay. These are the three substages during interphase. And M phase has two. Those are karyokinesis and cytokinesis. Okay. Karyokinesis has four substages. Those are prophase, metaphase, anaphase and telophase. Okay. So, here you can clearly see in G1 phase most of the organelle duplication takes place. In S phase, inside the nucleus DNA duplication takes place. Okay. And inside the cytoplasm centriole duplication takes place. In G2 phase, 3 organelles will duplicate. Those 3 organelles are chloroplast, mitochondria and Golgi bodies. These 3 organelles will duplicate in G2 phase, and tubulin protein synthesis also takes place in G2 phase. The interphase is very long and it is also called as inactive phase, where the cells are metabolically active, but it is not involving in division process. That is why it is called as inactive stage. Okay. So, yes, this is about cell cycle. Now, if you look into the M phase, that is karyokinesis, first it is prophase. In prophase, you can see the chromatids, thread-like structures called chromatids, and nuclear membrane, nucleolus will disintegrate here, where they lose their structural stability. And you can observe these chromatids clearly under light microscope during prophase. And these chromatids will come at the center, forming metaphasic plate like this. They will come at the center initially to the kinetochores of the each chromosome. Many a times questions asked based on this kinetochore, spindle fiber attachment. Spindle fiber will attach to the kinetochore of each chromosome, and during metaphase, okay, during metaphase spindle fibers are attached to the kinetochores, and that is how a single metaphasic plate is formed. Then coming to anaphase. In anaphase, what happens, Nana? Centromere splitting takes place. In anaphase, centromere splitting takes place. Centromere splitting will take place during anaphase. And then in telophase, again nuclear membrane, nucleolus will reappear. Nuclear membrane and nucleolus will reappear during telophase. Fine. And later cytokinesis will happen. Fine. So, these are different events of karyokinesis where you have to fix it in your mind. Next, when it comes to meiosis process, beta, in meiosis, prophase, meiosis 1 and meiosis 2 takes place. Okay. Meiosis 1 and meiosis 2. Keep each and every diagram in your mind. First, in meiosis 1, if you see, prophase 1 has 5 substages, beta. Prophase 1 has 5 substages. Those are leptotene, zygotene, pachytene, diplotene and diakinesis. Diplotene and diakinesis. So, in leptotene, what happens? Again, chromatids formation will happen. They will be attached to the membrane. This we will be calling it as pyreme stage. This we will be calling it as pyreme stage. In zygotene, synaptonemal complex formation, tetrad formation, bivalent formation takes place. When it comes to pachytene, these bivalents form recombination nodules. Okay, the homologous chromosomes, the non-sister chromatids. Do remember what are they? Non-sister chromatids of homologous chromosomes. Non-sister chromatids of homologous chromosomes. These non-sister chromatids of homologous chromosomes, they form recombination nodules. This crossing over takes place during pachytene. This is an enzyme mediated process, and the enzyme involved is recombinase. Okay, the enzyme is recombinase. Then here in diplotene, what happens, Nana? In diplotene, the dissolution of synaptonemal complex takes place and formation of X-shaped structures. Those are chiasmata, formation of X-shaped structures, chiasmata, that is formed in the diplotene. In diakinesis, terminalization of chiasmata takes place. So, these are the 5 events of prophase 1, so many times asked either in the match the following questions or statement based questions, very important, beta. So, here this diagram is implying to the third phase of this substage, that is pachytene phase. Next, metaphase 1, you can see bimetaphasic plate, that is 2 metaphasic plates are formed. Next, it is anaphase, where centromere splitting will not take place in anaphase. Keep it in your mind. No centromere splitting. No centromere splitting. No centromere splitting. No centromere splitting in the anaphase. Okay, the two chromatids in the homologous chromosomes, this will move towards this pole and this set of chromosomes will move towards the other pole, but there is no centromere splitting in the anaphase 1. In telophase 1, two daughter cells are formed, reforming back their nuclear membrane, nucleolus, and decondensation of the chromosomes into chromatids or this thread like structures. Next, coming to the prophase 2. In prophase 2, each daughter cell will again pass on to the same phase. Meiosis 2 is similar that of mitosis. Meiosis 2 is same as that of mitosis. In this stage, you can clearly see in metaphase 2 only single metaphasic plate is formed, and in the anaphase 2 chromosomes move apart, fine. Splitting of the centromere takes place in the anaphase of mitosis and anaphase 2 of meiosis, and at the end of meiosis process you can find 4 daughter cells, 4 haploid daughter cells, fine. Now, coming to the chloroplast structure, just now we have looked into it in the previous unit also, again now in the photosynthesis in higher plants. This is stroma, this is 2 membrane bound, and the fluid filled space inside is stroma, and the membranous sac like structures are what beta? Thylakoid. Stalk of this sac like structure is grana, fine. Inside that you can also find starch granules, and this ribosomes are of 70S ribosomes. We have also discussed the sites where all reactions are taking place, right? Just fix all these labelings in your mind, fine. And next, if you look into the action spectra and absorption spectra, which is given by T.W. Engelmann by the experiment conducted on cladophora, that is algal photosynthesis. So, here if you see Nana, absorption spectra, action spectra, both are given. So, here what is absorption spectra and what is action spectra? Okay, what is absorption spectra and what is action spectra? Absorption spectra will depict different pigments absorbing light at different wavelengths of light. Okay, different pigments absorbing different wavelengths of light is depicted in the absorption spectra. If you can clearly see 400 nanometers, 500, 600 and 700 nanometers. If you can see that clearly, Nana, this is chlorophyll A. First we will look into chlorophyll A. Chlorophyll A will show 2 peaks, one close to 440 and the other is around 680 nanometers. At these two regions, chlorophyll A is showing the absorption peak. If you look into chlorophyll B, around 480 nanometers, chlorophyll B is also showing the absorption peak. Then when it comes to carotenoids, carotenoids also show 2 peaks in between 400 to 500 nanometers. When you look into all the 3 pigments which are given in this spectra, the maximum photosynthesis is conducted by, or the maximum amount of oxygen is evolved by pigment, that is chlorophyll A, because this is absorbing light at 2 wavelengths, one at 440 and other at 680 nanometers. Fine. When it comes to action spectra, Nana, action spectra will depict that pigment which is absorbing maximum wavelength of light. Action spectra will show the pigment which is absorbing maximum wavelength of light, where the maximum amount of carbon dioxide release is on the y axis and wavelength is on the x axis. Action spectra and absorption spectra of chlorophyll A is coinciding. You can see clearly. This is the graph of the absorption spectra of chlorophyll A and this is of action spectra of chlorophyll A. Both action spectra and absorption spectra of which pigment is coinciding? Chlorophyll A. And then if you see, this is light harvesting complex which is present in thylakoid lamellae. This is present in thylakoid lamellae. Okay. So, here if you clearly see in the light harvesting complex, you can see all the accessory pigments, or antenna pigments. okay, accessory pigments or antenna pigments you can observe in the light harvesting complex, which has the pigments like carotenoid, xanthophyll and chlorophyll B. Okay. This absorbing light at different wavelengths, they will emit it to the reaction center. This is the reaction center, where in reaction center molecule present is chlorophyll A. So, all these molecules comes under light harvesting complexes, which are present inside the photosystems in the thylakoid lamellae. Okay, in the thylakoid lamellae, if you see, you can clearly come across the photosystems. In this photosystems, light harvesting complexes are present, where light harvesting complexes have accessory or antenna pigments, which includes carotenoids, xanthophyll and chlorophyll B. Okay. And then they will give it to the reaction center, that is chlorophyll A, which is absorbing maximum amount of light. This is the primary acceptor that will raise its electrons to the primary acceptor. And then if you look into Z scheme, okay, Z scheme which takes place inside the thylakoid lamellae. So, here from the light harvesting complex to the primary electron acceptor, you can see here primary acceptor. Primary electron acceptor in the Z scheme is pheophytin. Okay, the primary electron acceptor in the pheophytin is pheophytin. The primary electron acceptor from light harvesting complex is pheophytin. Okay. From this pheophytin, the 2 electrons are given to plastoquinone. From plastoquinone, 2 electrons are taken from plastoquinone. As 2 electrons are passed from pheophytin to plastoquinone, protons are taken from stroma. Okay, protons are taken from stroma, which forms PQH2, plastohydroquinone. Okay, this H protons are given to the thylakoid lumen and electrons are passed to plastocyanin. Okay, till here photosystem 2 is done. From plastocyanin, again electrons are excited to photosystem 1. Okay, till here this is photosystem 2 initially, and then again to photosystem 1. In photosystem 1, again from the light harvesting complex, the maximum wavelength of photosystem 1 is 700 nanometers, maximum wavelength of photosystem 2 is 680 nanometers. So, here from plastocyanin, these electrons are given to ferredoxin, then again 2 electrons are given to ferrous sulfur complex. Okay. From this ferrous sulfur complex FMN, and the ultimate electron acceptor is located towards the stroma side. This is one more. The ultimate electron acceptor is located towards the stroma side, that is NADP plus. Okay, it is what, Nana? NADP plus. NADP plus along with protons, it forms NADPH plus H plus. Clear? NADPH plus H plus. So, here if you can see in this diagram, as light is accepted in the form of photons inside the thylakoid lumen towards the inner membrane of the thylakoid towards lumen, photolysis of water takes place, and here electrons are excited. Primary electron acceptor is pheophytin of photosystem 2. This is the maximum wavelength of photosystem 2 is 680 nanometers, and the electrons are passed in this flow, and last electron acceptor of photosystem 2 is plastocyanin. This gives to the primary electron acceptor of photosystem 1, that is ferredoxin, and the ultimate last electron acceptor that is located towards the stroma side of the membrane, thylakoid membrane, that is NADP plus. Okay, whereby the enzyme NADP reductase will reduce the NADP plus to NADPH2 inside the stroma. Clear? So, this is all about the Z scheme beta, so much important. You have to keenly highlight what is the primary electron acceptors of each photosystems and what is the maximum wavelength of the light. Okay, maximum wavelength of this photosystem 2 is 680 nanometers, this is 700 nanometers, that you have to fix it in your mind beta. Okay, so this is all about the Z scheme. Next, coming to cyclic photophosphorylation. Under cyclic photophosphorylation, the electron transport will take place in a cyclic manner, where here the maximum wavelength of light is 700 nanometers, and this takes place in stroma lamellae, wherein first electrons are excited and these are accepted by ferredoxin, cytochrome, BCF complex, and later to plastocyanin, and from plastocyanin back to the primary acceptor. This will be the cyclic flow, that is why it is called as cyclic photophosphorylation, where only 2 ATP molecules will be released in this process. Fine. And then if you see the chemiosmotic hypothesis diagram, so much important, chemiosmosis, okay, chemiosmosis in this diagram. So, here from photosystem 2, after pheophytin, from plastoquinone, 2 protons are coming from stroma to the lumen. Okay. Initially by photolysis of water, already proton accumulation is present inside the lumen, and during this Z scheme from PQ again protons are entering from stroma to lumen. Whichever little protons are present in the stroma, those protons are reduced to NADPH by the NADP reductase. Fine. As such, the proton concentration in stroma is reduced and proton concentration in thylakoid lumen is high. Okay. This will generate a proton gradient. That is why chemiosmotic hypothesis need, this is mostly asked question, it needs an ATP synthase. Okay. An ATP synthase that has F0 F1 pump, okay, that is F0 F1 pump, and a membrane proton gradient and a proton pump Fine. These are the 4 elements that it requires, and here inside the membrane F0 is present and towards the stroma F1 is present. This is F0 F1 channel, that is proton pump, that generates 4 protons in one cycle, and where ADP which is present in the stroma forms ATP, is phosphorylated to ATP. This is how ATP energy rich molecule is formed inside the stroma of chloroplast, that is from chemiosmotic hypothesis. So much important, you have to remember it. Next, coming to the dark reactions or light independent reactions, this is from C3 cycle or Kelvin cycle. First is carboxylation, second is reduction, and third is regeneration. Fine. So, this carboxylation reaction, wherein carbon dioxide molecule is fixed, primary acceptor is RUBP, which is catalyzed by the enzyme Rubisco, world's abundant enzyme Rubisco, and this forms the primary acceptor. That primary acceptor is that primary acceptor is 3-phosphoglyceric acid. Okay, that is 3-phosphoglyceric acid. This 3-phosphoglyceric acid is reduced to 2 molecules of glyceraldehyde 3-phosphate, whereby 1 carbon dioxide molecule will use 2 ATP and 2 NADPH here. Okay, it will utilize 2 ATP and 2 NADPH, and then this from this 2 molecules of 3-glyceraldehyde, 1 molecule will be escaping for the formation of glucose. Rest 5 carbons will regenerate as RUBP, where it requires 1 more ATP for this. So, to fix 1 carbon dioxide, it requires on total 3 ATP and 2 NADPH to form 1 glucose molecule. To form 1 glucose molecule, it requires 3 into 6, that is 18 ATP, and 2 into 6, that is 12 NADPH. Clear, Nana? Understood? This is all about Kelvin cycle. And then this is C4 cycle. In C4 cycle, you can clearly see It has a Kranz anatomy where vascular bundles are covered by bundle sheath cells. That Kranz anatomy you can observe in C4 plants, which are tropical plants, wherein here the carbon dioxide is first converted to hydrogen carbonate by the enzyme carbonic anhydrase, and here you can see phosphoenol pyruvate fixation happens. Okay, fixed phosphoenol pyruvate is the primary acceptor of carbon dioxide that will fix by the enzyme peptase into 4 carbon compound, that is OAA. This is transported through plasmodesmatal connections between mesophyll cell and bundle sheath cell. Inside bundle sheath cell, this 4 carbon compound is decarboxylated to carbon dioxide and C3 acid. Here carbon dioxide will conduct Kelvin cycle, C3 is regenerated back to phosphoenol pyruvate. So, this is the complete diagrammatic view. You have to remember each and every process here. C4 cycle takes place in mesophyll cell, C3 cycle takes place in bundle sheath cell. Mesophyll cells have granule chloroplast, bundle sheath cells will have agranule chloroplast. So, here if you look into total energy beta, that is 5 ATP for 1 carbon dioxide, 5 ATP and 2 NADPH are required. For glucose, if you for the formation of 1 glucose, it is 5 into 6, that is 30 ATP, and 2 into 6, that is 12 NADPH are found. Okay, and then this is a limiting factor of light. Okay, limiting factor of light, where on the x axis rate of photosynthesis is plotted, where on the x axis light intensity is plotted, on the y axis rate of photosynthesis is plotted. Okay, just at 10% of total sunlight, the light comes to the saturation level. Only 1 to 2% from this 10% is used as incident light. Do remember, this is used, only 1 to 2% is utilized as incident light. So, here if you see first, as the light intensity increases, rate of the photosynthesis also increases, but further increase after the saturation point will not show any rise in the rate of photosynthesis activity. When it is saturated just at the 10% of total sunlight. Okay, and then if you clearly see the glycolysis process beta, here what you have to remember, at which steps the energy is utilized and what are the enzymes involved in the each and every step. All these 10 biochemical reactions are important. Okay, glycolysis, which is also called as EMP pathway, as it is given by Emden, Meyerhoff and Parnas. This takes place in the cytoplasm, and this is common both for the aerobic respiration and anaerobic respiration. So, if you look into the first 5 steps, come under preparatory phase. Okay, first 5 steps comes under preparatory phase, next 5 steps comes under payoff phase. Okay, next 5 steps comes under payoff phase. At the first step and third step, and at the first step and third step, 2 ATP molecules are utilized. Okay, and these are rate limiting steps. The rate limiting step of glycolysis is third step, and the enzyme which is catalyzing this is phosphofructokinase. Okay, this is the rate limiting step. So, 2 ATP molecules are utilized. Okay, 2 ATP molecules are utilized in the first and third step. 2 ATP molecules are utilized in first and third step. Okay, fine. After the formation of this glyceraldehyde 3-phosphate, you can clearly observe at 6th step energy is released. Okay, at 6th step 2 NADH is released at the 6th step, where we have to count it for 2, as here glucose molecule is splitted into 2 3 carbon compound. for one glucose molecule in the 6th step 2 NADH is released. In the 7th step, if you see, in the 7th step and 10th step, on total 4 ATP molecules are released. From this 4 ATP molecules which are released into the cytoplasm, 2 are being taken. So, the net ATP, when it comes to the energetics of glycolysis, net ATP is 2 ATP, okay. Net ATP is 2 ATP, total ATP is 2 NADH into 3, 1 NADH is equal to 3 ATP. So, 2 NADH into 3 is 6 ATP, okay. 6 ATP plus 2 ATP is 8 ATP, the total energy is 8 ATP molecules, okay. And then if you see Nana, fermentation process, okay, once the phosphoenol pyruvate that is turning to pyruvic acid which is formed at the end of glycolysis, if it is conducting anaerobic respiration, 2 steps can happen. One is lactic acid fermentation, where it occurs in the muscle cells in the eukaryotes and in prokaryotes usually. This is called as lactic acid fermentation. In the 6th step, whichever NADH is released into the cytoplasm, that is utilized during lactic acid fermentation and alcoholic fermentation. So, that is 0 energy which is formed, the energy is 0 which is formed during the fermentation process, okay. So, for anaerobic respiration, the total energy released is just 2 molecules of ATP. The total energy released during anaerobic respiration is just 2 ATP, okay. 0 NADH is we are not counting, because the one that is released into the cytoplasm is utilized by lactic acid fermentation and alcoholic fermentation, clear. So, here at the end of alcoholic fermentation, ethanol and carbon dioxide is formed. Carbon dioxide is formed at the end of alcoholic fermentation only during anaerobic respiration. Keep that in your mind, okay. These are the major important points beta. Do remember where the energy is released in the 6th step of glycolysis, where is it utilized? During lactic acid or alcoholic fermentation. Keep it in your mind, fix all the terms and energies, Nana, most important. When it comes to TCA cycle or Krebs cycle, it is very important for you people to remember all the steps of this TCA cycle, okay. So, here the primary acceptor is OAA and this accepts acetyl CoA enzyme. By the enzyme citrate synthase, citric acid is formed, which turns to isocitrate, and isocitrate will undergo decarboxylation and the release of 2 NADH. We are counting for 1 glucose. 1 glucose means 2 TCA cycles will happen, okay. 2 TCA cycles will happen, okay. In that case, here 2 NADH and alpha ketoglutarate to succinyl coenzyme, again a decarboxylation and again 2 NADH. Then from succinyl CoA to succinic acid, 2 molecules of GTP, succinate to fumarate, 2 molecules of FADH2 and then malic acid to ascorbic, malic acid to oxaloacetic acid to NADH2. So, here this all steps come under oxidation steps, okay. So, here you can clearly see at 3 points totally 6 NADH molecules are released out for 1 glucose molecule, that is 2 cycles of TCA and 2 molecules of FADH2, 2 molecules of GTP are released, okay. Keep that in your mind, all the steps are important beta. And next, if you see electron transport system, most important question, highly important question, you have to look into it. All the 4 complexes, the very first complex is NADH dehydrogenase, complex 2 is succinate dehydrogenase, complex 3 is cytochrome BC oxidase, complex 4 is cytochrome C oxidase, complex 4 is what, Nana? Cytochrome C oxidase. This is complex 4. So, here in the complex 1, if you see NADH dehydrogenase, NAD plus is where NADH is again oxidized to NAD plus, and the protons are pumped to intermembrane space. At one turn 4 H plus are released and 2 electrons are given to 2 protein molecules. First is flavin mononucleotide FMN, then the protein is ferrous sulfur complex. 2 electrons are given. That is given to the mobile electron carriers. Next, electrons are pumped to mobile electron carriers, that is ubiquinol from complex 2, which is not transpanning the 2 sides. Here both the electrons and protons are submitted to ubiquinol only. Here complex 3 again true ferrous sulfur complex and mobile electron carrier, that is cytochrome C, the electrons are passed to complex 4. Only in complex 4 you can see copper ions along with cytochrome A and A3. At the end you can clearly see the ultimate electron acceptor in the ETS is oxygen. Okay, the ultimate electron acceptor is oxygen, and this complex 4 at the end it will form water molecule, okay, where it is called as cytochrome C oxidase. Fine, all this will generate proton gradient again in between intermembrane space and matrix. So, the protons from the intermembrane space are moving into the matrix and finally leading to the formation of ATP and making the mitochondria the powerhouse of the cell. So, this is all about the electron transport system that you have to remember beta. And next, this is the protein pump channel, wherein F0 is inside the inner mitochondrial membrane and F1 is towards the matrix. This you have to remember clearly, the differentiation, where ATP synthase is inside the chloroplast. Inside chloroplast F0 is in the membrane of the lumen and it is towards the stroma, right, where in the chloroplast, if you see chloroplast, okay, F0 is inside the thylakoid lamellae and F1 is towards the stroma, okay. But here you can see F1 is towards the matrix and F0 is towards the inner mitochondrial membrane. That you have to keep it in your mind. And then the respiration is called as amphibolic pathway because all the respiratory substrates will have one common cycle, that is Krebs cycle here, okay. All the respiratory substrates will involve in the same pathway only. For example, and here both the anabolism and catabolism are involved. Like fats, first they will break down to fatty acids and glycerol, okay, they will break down to fatty acids and glycerol. Fatty acids will turn to acetyl CoA, fine, and proteins will first lyse to amino acid will join as pyruvic acid. So, Krebs cycle is common pathway for all the respiratory substrates. And here where do you see the anabolic processes? Anabolic processes you can find out like acetyl CoA might be, succinyl CoA in the TCA cycle might be, alpha ketoglutaric acid, all these organic intermediates act as the starting molecules for many biosynthetic processes, especially the biosynthesis of secondary metabolites. So, as both anabolism and catabolism are involved, we will call it as amphibolic pathway. Here what you have to remember, Nana, which like proteins are joining as pyruvic acid, fats from fatty acids to acetyl CoA. This is more important. Keep labelings in your mind all the time. Now, the hypocotyl seed germination, where hypocotyl radical is first formed into root and then the shoot formation, the epicotyl hook will be germinated. So, this hypocotyl seed germination you will observe in mostly pea, bean, that is Fabaceae family. And here you can clearly see the elongation of the root and shoot, okay, elongation of the root and shoot, and at the central part you can see the vascular cambium. At the tip here you can see shoot apical meristem and down here you can see root apical meristem. And then the arithmetic and geometric growth. Arithmetic growth you can see, arithmetic growth you can clearly see in the root elongation, arithmetic growth you can see in the root elongation process, wherein one cell will divide into two, of that one cell gets permanent and other is meristematic. Okay, this permanent cell will have its functional role, and this meristematic cell will again undergo division, of that one cell gets permanent and one is meristematic. Geometric growth rate, if you see, one cell dividing into two, all the cells will undergo cell division and further it will increase the cell number. Best example is bacterial culture. Here you can see arithmetic growth rate will give you a linear curve, and the formula is length LT is equal to L naught plus LT is equal to L naught plus RT. Okay. This is the formula for arithmetic growth rate, where linear curve is seen from y axis is on the height of the plant and x axis time, LT is equal to L naught plus ERT. Whereas sigmoid growth curve, S-shaped curve or sigmoid curve you can see in the, that is the exponential growth that you will see in the geometric growth phase, where you can observe three phases here: lag phase, log or exponential phase and stationary phase. On the y axis you can see size or weight of the organ, and on the x axis you can see time. And here the formula for geometric growth rate is W1 is equal to W naught ERT. Fine. The labelings are again important. LT is the length of the plant, final length, initial length, growth rate, time. Okay, final weight, initial weight, this is E is the base of the logarithm, R is growth rate and T is time. Here you see absolute growth rate and relative growth rate, where qualitative comparison you can see between absolute growth rate and relative growth rate. Absolute growth rate formula is what, Nana? Final size minus initial size. Okay, relative growth rate formula is final size minus initial size by initial size into 100. Okay, keep this formula in your mind, very much important. And then this you can see different developmental pathways from meristematic cell. Plasmatic growth means growth in the size of the cell, be it cell organelles, be it cellular constituents, and that gets differentiated to a mature cell, which finally undergoes senescence and leads to death. Okay, so meristematic phase, elongation phase, maturation phase and finally death. Okay, and here what is plasticity? Different developmental pathways followed by the plant and different structures you will see in the same plant. Either it can be through phases of growth or based on the environment they will grow. Okay, one best example is heterophily. Heterophily based on the phases of growth, you can see like juvenile phase one structure and adult phase one structure. Best example for the phases of growth heterophily are, best examples are cotton, coriander and larkspur. Cotton, coriander and larkspur, wherein this if you see based on the environment, different different structures of the leaf based on the environment, terrestrial and water habitat. Best example is buttercup. Sorry. Best example is buttercup. Yes, with this we have finished all the diagrams of NCERT 11 beta. Any doubts, anything you have, you can drop it in the comment section. I will be answering all your doubts, and once have a rough look on all these diagrams, you can quickly go through the session, and in the next session we are going to discuss by today evening like 5 o'clock, we are going to discuss the diagrams of NCERT 12, and quickly we will practice few objective as well. Okay, fine. All the best for your exam.